Welcome folks. In this video we are going to look at biology paper 2 for the year 2002, and this paper was written by the internal candidates. So we have question 1. Figure 1.1 shows tissues from organisms. Okay, so we have tissue A, tissue B, another tissue D, and the structure C. Now let's have the first question here. Question A1, name tissue A. So if you look at the tissue A, this region is where the palisade cells are found. So the question is the same as what tissue can be formed or is formed by a group of palisade cells. So this tissue is known as palisade mesophiles tissue. Then the second one, we are looking at tissue D. So these are ciliated cells. So in which tissue or tissues are ciliated cells found? So these are known as ciliated epithelial tissue. Okay, so we move on to Roman number 2. Where do you find tissue C and D? So now if you look at the tissue C, we are saying the tissue C, this should be an organ, and this is the leaf. Now the leaf has several functions, among of which we have the production of glucose by the process of photosynthesis. Now tissue C, which is the organ, which is the leaves, is found. So this is the leaves, and to specify this, it is found in plants. Then for tissue D, this is the respiratory tract. Respiratory tract, or sometimes, not sometimes, you can also find it in oviduct. So we can just say in animals as well, such as human beings. We have the oviduct where the process of fertilization is mostly likely to occur. Then if you look at the leaves, we are saying we have the production of glucose by photosynthesis. Then we also have transpiration. Transpiration is the loss of water through the leaves. Now, transpiration may be the loss of water, but it is a vital process as it aids the nutrients to move from the soil up to the leaves. Then also in cooling of the temperature of a plant. Then if we move on to question B, describe how the tissues C and D are adapted to their function. So tissue C, we are looking at the leaves. So adaptation. Okay, so now let's just focus on leaves in terms of the process of photosynthesis. So we'll say this is where the process of photosynthesis occurs. It is due to the presence of palisade cells which contains chlorophyll. And we know whenever we are dealing with the process of photosynthesis, whenever we look at chlorophyll, it is a green pigment that traps sunlight. So the presence of chlorophyll helps the process of photosynthesis to occur. Then the reason behind why the process of photosynthesis is very high in palisade cells, it's where we have a highly concentrated of the chloroplast. And we know that the chloroplast is the site where the process of photosynthesis occurs. Now, if you look at structure D, we remember structure D, these are ciliated cells. So what adaptation can we assign to ciliated cells that helps the ciliated cells to do their work? So we'll say these cells contains contains hair, contains hair-like structure. Called cilia. So say these traps dust particles and helps the air to move in one direction. So this is what we can say about this. So remember, if you look at ciliated cells, we are definitely looking at some of the parts that are found along the respiratory tract, and their function is to remove the dust particles, because what we want is by the time the air reaches the lungs, which is where the diffusion of gases or the exchange of gases occurs, we need the air by that time to be clean so that we avoid some respiratory diseases. So that is what we can assign to that. So now let's move on to question 2. Under question 2, we have figure 2.1 shows sections of blood vessels R and S. Now, blood vessels, we are dealing with circulatory system, and under circulatory system we have the lymphatic system and the cardiovascular system. Now, the blood vessels, these are structures that are found in a cardiovascular system, and we have 3 types of blood vessels, among of which we have the arteries, and we need to look at the structure of the artery, then also the function of the artery. Then we have the second one, the vein, the structure, then also the function. The third one is the capillary. Now, from first question, A Roman number 1, name the type of blood vessels shown in figure 2.1. Blood vessel R. So looking at the lumen, so the lumen is just this hole found in the middle. So this is what we call the lumen. Then the muscles here, the muscles, we have the muscles. So if you look at this thickness compared to this one, so we can tell and see that blood vessel R is thicker than blood vessel S. Now, looking at the size of the lumen, we can see that it's very big. So vessel R is a vein. Then for S, we have a capillary. So capillary is easy, our S. Now for an artery is a blood vessel with the smallest lumen. It is very small. So the arteries are blood vessels with very thick muscles, and the reason behind that is because the arteries carry blood at high pressure. So for pressure to be created, we need muscles that are very thick. So remember that. Then if we look at Roman number 2, describe structural differences between the blood vessels R and S. So structural differences is, we are not going to look at the functioning, no, but we need to look at the difference in terms of the structure, the appearance. What can we see between the two? So the first difference that I can spot here is that blood vessel R has thick muscles than blood vessel S. Then in terms of functioning, the blood vessel S, which is a capillary, connects the artery to the vein, or it is a vessel that does the completion in terms of the distribution of nutrients to the cells and also the removal of some of the waste products. Then away from that, let's go to question B. Two patients went to the hospital on suspicions that they had malaria infection. Their blood samples revealed the following. Patient X had no malaria parasites but was anemic. Now, anemic is a condition among of which we may have, it may be caused by lack of iron. I mean iron, not iron. Then the second one, it can be lack of vitamin B12. Okay, then let's look at patient Y. So patient 1 had malarial parasites and was anemic. So two conditions in patient Y. Roman number 1, explain how malaria can cause anemia. Now, anemia is a condition that can arise from lacking of these nutrients. Apart from the deficiency, it can also be caused because of a malaria parasite. Now, if you've studied carefully, you've looked at a plasmodium. So plasmodium is a pathogen that causes malaria. Don't confuse a plasmodium with a mosquito. A mosquito acts as a vector because it transports the plasmodium from one individual to another individual. Now for plasmodium, so what happens is that whenever the person who is affected with the malaria, the plasmodium will infect or will attack the red blood cells. From there, the red blood cells will become ruptured, and because of the rupturing of the red blood cells, we may now result in an anemic condition. So say the malaria parasite can infect the red blood cells, resulting in rupturing of the red blood cells. So that is how malaria can cause anemia. So by just the results on, whenever this parasite of Plasmodium is injected in blood, its target is the red blood cell. So after it enters inside the red blood cells, it is going to undergo some life cycle. After the life cycle, then the red blood cells will be ruptured. From that, then we may have the condition of anemic. Then Roman number 2 states other possible causes of anemia in patient X. So the first one, we can look at lack of iron. Then the second one, we've looked at that one, can be lack of vitamin B12. So we move on to Roman number 3, patient X with blood group A needed blood transfusion. Name a possible donor. So we have 2. So it can be blood group A or O. Then not only are we supposed to look at this, we need to make sure that even if the donor has blood group A or O, we have to make sure that the blood that we are receiving must not contain any anemic condition. So we need a non-anemic condition. So that one must be put into consideration. So the point is, we can't transfuse blood with anemia or with such a condition from one individual to someone who has anemia. So you need to get that into consideration. So we can say a person must not have anemia. So that also is very important. All right, we move on to question 3. Figure 3.1 shows two cross sections from different parts of a dicotyledonous plant. So examples of dicotyledonous plants, we have like a bean plant, we have mango plant. All these are examples of, and under the structural appearance, you should be able to see the difference in terms of the root and of the stem. So we have plant part W, the first one, then plant part V, the second one. Now the question is, which diagram shows the cross section of 1 root, number 2 stem? Explain your answer. So how do we identify the structure of a root of a dicot? So we just have to look at in the middle. So the shape of the xylem, we can see that it is the first one in the center, the second one it is a star shaped. So that's the reason. So the root is, we just say plant part W. Reason, we can say because the xylem is located in the center and star shaped. So that's what we can use to identify a dicot root. Then for a dicot stem, we can see these, they are arranged in a ring. And if you look at the monocot stem, the way the vesicular system, which is the xylem, phloem and cambium, they are located like they are just randomly. There is no order. They are not located or arranged in a ring like structure. So that's what you need to use for you to identify such a part. So stem, plant part V. So we'll say because the xylem, cambium, and phloem are arranged in a ring like pattern. So that's what we can use. Okay, so we move on to row number 3. What are the shaded regions in the diagram? Give the reason for their different distributions. So the shaded region, if we go back, so these are the xylem. Okay, now for the question, the reasons. So the reason why they are shaded, because they transfer a solution. They transfer solutions which contains salt, which is salt. We are looking at ions, salt and water, from the soil to the leaves. Why to the leaves? Because it's where the process of photosynthesis occurs. Okay, now we have question B. Herbaceous stems and wood stems were clamped in water. Figure 3.3 and concentrated solution, figure 3.4 are shown below. So the first one, we have 2. So let's not focus necessarily on these stems. We know that these are plants and they are made up of the cells of the plants. Even here we have the cells. So this question is same as what happens if you place a plant cell in water. So this is same as diluted, or we can just consider it as pure water. So The concentration of salt solution in cells of herbaceous and wood stems is much greater than that of water. So it means the water molecules will move from here into the cells. So that is the observation. Now for this case, these two cells were put in salt solutions. So this time, the water molecules will diffuse out. And as the result, the mass may reduce, including the shape. But for the first one, the mass may change. Then we have described what will happen in figure 3.1 and figure 3.2. Explain your answer. So figure 3.1, the cells will gain water. Will gain water through osmosis. Explanation. So say this is because water molecules will move from the surrounding into the cells of the plants. So we'll move from the water surrounding. So the water surrounding is considered to be of high water molecules in terms of concentration. So it means there is less water in the cell in the cells. So the water must move from where there is in a high concentration into where there is a lower concentration. Then for figure 3.2, the case is different. So say the cells will lose water. So we'll lose water into the solution and shrinks in size. Remember, for the first one where the plant cells gains water, they do not burst because of the presence of the cell. But for the animal cell, we may expect to have a cell crenation. So now explanation, because water will move from the cells into the solution where there is less water molecules. Okay, so we move on to question 4. Question 4, figure 4.1 shows the changes in size of a population of producers and of a population of consumers in a lake over several months. So we have been given the graph. In the y-axis we have population, the x-axis gives us the month. Then in the first question A is what is meant by producer in a food chain. So the producers are always the green plants. So the producers are just living organisms that have the ability to produce their own food by using the sunlight. So say organisms with the ability to make their own food. Their own food through photosynthesis. Okay, so we have B, which graph X or Y represents the consumers? Explain your answer. So the consumers are organisms that depends on the producers. So we have the primary consumer, secondary consumer, but let's not focus on those. So we want to look at which one can we look at as the consumers. So the consumers is Y, because at the beginning of the food chain, the producers are always in a greater number. So this is a consumer. Then we have a producer. So the producers, the plants, the population of plants is greater than the consumers, like animals. Let's say, for example, human beings. The number of human beings cannot be compared to the number of plants. There are a lot of plants compared to animals. So that's what we can. So we can see that here, this is greater than this point here. So it's the consumer, graph Y. Explanation, because there is a greater population there is a greater population of producers, but less population of consumers. Okay, we move on to question C. The change in population size of the producers between the month of June and September was a result of farming around the lake. Suggest how farming methods may have caused this change. So let's have a look at June and September. So here, September is here. So let's look at at this point, the producers has gone up. Then the consumers have gone down. So there may be some reasons. So since we are looking at the month of June and September and also of farming, so what can cause the producers to go up in terms of farming? It can be application of fertilizer. We know that whenever we apply fertilizer to plants, there is that rapid growth of plants. So the application of manure and fertilizers. So this is what may or can cause such. Then we have question D. A biologist studying the lake several weeks after September found that the size of both populations had fallen greatly, while the number of bacteria were very high. What term is used to describe this effect? So the effect where there is an increase of bacteria is called eutrophication. Eutrophication. So what are the advantages? So disadvantages over increase of the bacteria population. Then also, if there is the over increase of bacteria, let's say in water, these bacteria may use the oxygen that is found in water, and whenever the oxygen is used up completely, then the aquatic living organisms such as plants, including fish or animals that are found in water, may starve or die because of the absence of oxygen. So we can say over depletion of oxygen. So sometimes also it may sometimes over increase in aquatic plant. Sometimes we may have a condition where there is too much plants in water, and because of that the plants are using too much of that oxygen in water. As a result, there is less oxygen for other organisms to survive, because if you look at the nature and the ecology, it tells us that every organism depends on the other one. So now he explained the relationship between graphs X and Y. So the relationships that the population of consumers depends on the population of producers. So that's what it depends. We need producers for this to survive. Then the last question, question 5 for genetics, we have a farmer carried out a research by crossbreeding a white corn variety and a brown corn variety. Most of the cobs that have developed had white corns. The farmer then planted some of the white seeds, batch 1, and the plants were allowed to self-pollinate. So we have self-pollination. Most of the cobs that developed from these crops, we call these as batch 2, had a mixture of brown and white corns on them, as shown in figure. Okay, so we have a maize cob here. The farmer randomly collected 5 cobs and counted the corns on each cob. Okay, let's have the table. So table 5.1 shows the number of corns from each of the 5 cobs. Okay, that's the case. Then the first question, calculate the total number of corns of each color on table 5.1. So we just have to do some math. So begin with the white corns. So the first cob 121, 21 plus 17 will give us 38. 38 plus 52. Let's just get a 2. 2 plus 38 is 40. 40 plus 50, 90. 90 plus 30, that is 120. 120 plus 36 is 156. So here we have 156. Then number of brown corns, 6 plus 7 is 13. 13 plus 17 is 30. Plus 11, 41. Plus 11, 52. 0 plus 0 plus 0 plus 0 plus 1 is just 1. Problem number 2, what is the ratio of white to brown corns? So the ratio, white is 156 to 52. Then if you want, you can reduce. We can divide by 2 into 156 will give us this should be 78. So 78, then here we have 26. We can still reduce. So into 78 we'll have 30, should be 38 to 13. Then which color is dominant? So the one that is dominant is the one that is easily C, the one that has the power to overshadow the other 2 colors. So from the 3 colors that we have, the one that shadowed the other 2 colors is white. So white was dominant. So say white color. Okay, now we have Roman number 4. Using LOS B and B, a genetic diagram, explain how the seeds from batch 1 could give such a result as shown in table 5.1. So we'll first begin by parental. So we'll start with parental phenotype. So the first one, we know that whether male or female, it was batch 1 was white. Even the other one, since it was self-pollinated, we only have white. Then we look at parental genotype. So since genotype. So since these were carriers, we just have to put capital letter B for white, small letter b for brown. So that's what we are going to do. Then from there we'll say parental gametes. So parental gametes, we just have to separate these. Then from there we can have random fertilization. So this one, heterozygous for white. This is white but heterozygous. Here we have homozygous for white. Here heterozygous for white. Heterozygous simply means having, okay, there is a mistake that I've made. I've made a mistake here. So this one must go with this one. So homozygous for B, which is white, pure white. Heterozygous, here a carrier, heterozygous, then homozygous for. So these are genotype of first filio generation, which is the generation for offsprings. So let's look at the chance. We have chance number 1 white, number 2 white, number 3 white. So here is brown. So from the four chances, the chance to have white is equal to 3 over 4 times 100%. And if we do some math here, it is going to give us 75%. So 75% to give us white, then 25% to give us brown. As a result, that is why we have a greater number of white cobs. Then we move on examining the cobs. The farmer noticed that cob 5 had one cone which was purple colored. Suggest the process which most likely brought about purple color in cob 5. So this can be variation. So variation may be the cause. Now, assuming the allele for purple color in this cob is recessive, briefly explain how the farmer would obtain many. So by crossing homozygous purple, so purple maize. So whenever we want this, we just have to make sure that we are crossing the homozygous. So that's it for this video. Thanks for watching.